So I'm going to tell you basically about a technology development project towards trying to create some protein-based reporters of membrane voltage. So when a neuron expresses these proteins, action potentials, spikes in the membrane voltage, are accompanied by flashes of fluorescence that we can see in the microscope. And this is real data, and that's really the whole story. Before I take questions, I'll go into a little bit more detail. And by way of disclosure, I started a company which is working on developing some of this technology. So this is the artist's conception of what we want to do. We want to make neurons light up when they fire so that we can see their underlying electrical activity. And this isn't a new problem. People have been working on this for decades. The challenge is that if you just look at the neuron, there's no endogenous contrast, which tells you about the voltage. So we need some molecular tool to convert these relatively small and brief changes in voltage into an optical signal that we can see. The story I'll tell you starts here. This is a view from an airplane looking down. This is the base of San Francisco Bay. There are salt ponds here where people are evaporating seawater to make salt. And there are these beautiful colors in the water. To see where these colors come from, we can zoom in by 12 orders of magnitude. And what we see is that there are microorganisms growing in the water. And in the membranes of these microorganisms, there are these proteins called microbial rhodopsins. They're transmembrane proteins. Covalently bound in the core of the protein is this chromophore called retinal. It's a conjugated molecule. And it's this chromophore which gives the color that you can see from the airplane. There are thousands of varieties of these proteins known in the wild, more than 5,000 varieties. And they come in all the colors of the rainbow. Here are some of the ones that we've worked with in my lab. In fact, this is a little bit counterintuitive because they all have exactly the same chromophore in them. It's this molecule of retinal, which is the same in all of them. It turns out that the absorption spectrum of the retinal is very sensitive to the electrostatic environment that's set around it by the protein host. The same situation applies in our eyes. So the same chromophore, retinal, is the only molecule involved in color vision. But most of us have color vision, and this is because different opsins in different cone cells in our eyes tune the absorption spectrum of the protein to different parts of the visible spectrum. So in a sense, the color tunable range of these proteins defines the visible spectrum. Now, these proteins mediate a huge variety of interactions between sunlight and biochemistry. So for instance, there are light-driven proton pumps, there are light-driven chloride pumps, light-activated ion channels, and light-activated signaling molecules. And of course, famously, everybody here knows that about a decade ago, people realized that you could take the genes for these proteins and introduce them into other cell types and thereby gain optical control over membrane voltage in these other cells. And so the most famous class of these are the channel rhodopsins, which can depolarize a neuron with light and cause it to fire. And this has led to great advances in neuroscience, which we all know about. So here we have these proteins where light comes in, and it leads to a change in the membrane voltage across the cell. So a few years ago, I said to myself, I wonder if we can run these proteins in reverse. Instead of having light come in and a change in voltage come out, can we use a change in the voltage to produce an optical signal that we can detect? Okay? So instead of light in and voltage out, we want voltage in and light out. So now I come here to Israel. It turns out there's actually a local piece of history to this story. So everybody here should know where this is. This is Yam HaMelech. This is the Dead Sea. And yesterday I met with Aharon Oren, who's a professor here at Hebrew University, who discovered the protein that I work with. And he told me a little bit of the history, which I'll just share with you. So the story started in 1979 during the Arab oil crisis. And Israel was trying to figure out a way to reduce its dependence on foreign oil. And so there was discussions to make a conduit from the Mediterranean to the Dead Sea to generate hydroelectric power from the difference in water pressure. And so somebody thought, well, maybe we should look to see what's in the Dead Sea, to see what happens if we dump huge amounts of Mediterranean seawater in there. And so they commissioned Aharon to do this, and he looked in the Dead Sea, and saw that when the water became slightly diluted, there was a bloom of a red archaeal microorganism called Halorubrum sodomense. This microorganism is red because it produces a protein called Archaerhodopsin-3. This protein is a light-driven proton pump. So it absorbs green light, and it uses that energy to pump protons from inside the cell to outside the cell. This establishes a proton motive force, which this creature then uses to power its ATP synthase to make ATP. So it's a way of making a living from sunlight. It's a primitive form of photosynthesis. So that was 1983. My lab discovered that this protein is a little bit fluorescent. It can be excited by red light, and it emits in the near infrared. But it's only fluorescent when there's a proton sitting on a particular functional group right here in the middle of the protein. Now, this proton is charged, and so if there's a change in the membrane voltage, that transmembrane electric field can push that proton onto the protein or pull it off. So you can tune the location of the proton, you can tune this acid-base equilibrium with membrane voltage, and that can regulate the fluorescence of the protein. So we excite the protein with red light, and then when we apply a voltage, the proton goes on and the fluorescence comes out. Right? And you see this works in PowerPoint, and so that's very encouraging. Okay. So we took the wild-type protein straight out of the Dead Sea, and we expressed it in a HEK cell. So this is just a human tumor cell. And we're exciting it with red light, and we're looking at the near-infrared fluorescence. And you can see, because it's a membrane protein, most of the fluorescence is localized to the periphery of the cell. You can't see it in the picture, but there's a little patch pipette stuck in the back of the cell. And I'll show you a movie here where we're applying steps in voltage between plus and minus 1/10 of a volt, and we're just looking at the near-infrared fluorescence of the cell. What we see is that when the voltage goes up, the fluorescence gets bright, and when the voltage goes down, the fluorescence gets dim. Okay. Now I'll tell you a little bit about some of the technical properties of this protein. So we excite it with orange or red light. It emits in the near-infrared. This is very convenient for biological imaging because it leaves the rest of the visible spectrum open, and there's little autofluorescence at these wavelengths. The quantum yield is terrible. It's about 1 or 2%. So this means for every 100 photons we put in, we get about 1 or 2 photons back. So it's very dim. But the low brightness is partially offset by the fact that this protein is very photostable. So here's a photobleaching trace showing a typical microbial rhodopsin and an organic dye under the same illumination intensity. And you can see the dye photobleaches in about 30 seconds, while the rhodopsin lasts about 30 minutes. So even though these proteins are dim, because they're in the near-infrared and they're so photostable, we can turn up the laser power and still get reasonable amounts of light out of these proteins. Now, in the wild, this protein acts as a light-driven proton pump. So shining light on it passes a current, or leads to a current, which then perturbs the membrane voltage. And we're trying to make a sensor, not an actuator. So we don't want to disturb the quantity we're trying to measure. Here's the wild-type protein. When we turn the laser on and off, you see we get this photocurrent. It turns out it's relatively easy to mutate the protein to block its function in the forward direction, but to still preserve its reverse function as a fluorescence voltage indicator. And so by mutating one of the residues in this proton transport chain, we made a version which doesn't show a photocurrent. Then working with Robert Campbell's lab at the University of Alberta, we engineered other variants which are faster and brighter and more sensitive, but conceptually the same. Now, an important figure of merit for a voltage indicator is its sensitivity. How much change in fluorescence do we get per change in voltage? To measure that, we express the protein, and then we apply triangle waves of voltage, and we plot fluorescence as a function of voltage. The wild-type protein has about a 50% change in fluorescence over a physiological range. Our engineered mutant, our best one, which we call quasar 2, has about a 2-fold change in fluorescence over a physiological range. The other important parameter is the speed of the response. To detect an action potential, the response has to be in a millisecond or less. And so we apply steps in voltage and plot fluorescence as a function of time. And both the wild-type protein and this mutant have a dominant response in less than a millisecond. The mutant also has a minor component which has about a 15 millisecond response time. All right. So now let's look in neurons. We started in cultured rat hippocampal neurons. So here's a neuron in a dish. You can't see it in the picture, but there's a little patch pipette stuck in the back of the cell. And I'll show you a movie of an action potential in the cell when we trigger it to fire with electrical pulses from the patch pipette. This movie is taken at 1,000 frames a second. We have to image very fast because the signals we're after are so brief. And so this is what a neuron looks like when it fires. Okay. So you can see, you can actually see the electrical signal in the cell. By averaging over the whole cell, we can extract the fluorescence as a function of time, and that's shown in red here. This is single trial data. This movie is averaged over about 100 repetitions of the experiment to improve the signal-to-noise ratio. Here's a close-up, and you can see the correspondence of the optical signal and the electrical signal. Just to compare this to calcium imaging, which is, of course, much more familiar, we made a construct where we could simultaneously image voltage and calcium. We just stapled a genetically encoded calcium indicator, a GCAMP, to the voltage indicator. The GCAMP indicator is blue, the voltage indicator is red, so we can look at both at the same time. We expressed it in a neuron and then recorded both channels. And you can see that here's the voltage signal, here's the calcium signal. And so while they both report activity, the voltage signal gives the spike timing much more precisely and can report fast bursts of spiking. Also, subthreshold events like this ePSP here show up in the voltage signal but have no correlate in the calcium signal. Inhibitory events also show up in the electrical signal when they occur. Okay? So we call this fusion construct for the calcium and voltage indicator. We call it caviar. Now, there I was showing you spontaneous activity. And before that, I showed you an example where we were using a patch pipette to stimulate the neuron to fire. But using a patch pipette to stimulate the cell partially defeats the purpose of having this nice optical readout, because you still have to go and poke every cell to stimulate it. But we all know that there are these channel rhodopsins, which you can use with light to stimulate a cell. So I thought, why don't we just co-express a channel rhodopsin and a Archaerhodopsin-based voltage indicator in the same cell. That way we can stimulate the cell with blue light, and we can record from it with red light. So working with Ed Boyden's lab and Robert Campbell's lab, we did this. It turns out that the channel rhodopsin 2 that is most commonly used, its action spectrum is slightly tickled by the red light here, and so there's crosstalk. And so we developed a new channel rhodopsin variant derived from a freshwater alga, which lives in a pond in the south of England. It's called Sheriff. And this channel rhodopsin is, it's similar to channel rhodopsin 2. It's slightly blue shifted, and it's about 10-fold more sensitive. But otherwise, it's the same idea. So we'll flash the cell with blue light, and that will trigger it to fire, and we'll see the response as a flash of red fluorescence. Okay? Like that. And so here's real data. This is, again, on a rat hippocampal neuron in culture. Each blue bar represents a flash of blue light delivered to the cell. The red is the single trial fluorescence recording, and the black is the simultaneously acquired patch clamp recording. Here's a close-up, and you can see the correspondence of the optical and electrical signals. But now we don't need the patch pipette for at least these in vitro measurements. Here we're just using it for ground truth to calibrate. Now, one of the neat things about having a genetically encoded system for optical stimulation and recording is that you can use genetic tricks to control which cells have the light-gated actuator and which cells have the voltage-dependent reporter. And so a student in my lab developed a system for specifically probing synaptic transmission in cultures. And here's how it works. We deliver the channel rhodopsin to all the cells in high titer, so they all start to express the channel rhodopsin. And we deliver a flexed version of the voltage indicator. It's called quasar. This is 3 now. The numbers will keep getting higher as we improve them, but it's the same idea, of the voltage indicator. So it doesn't express in any of the cells. Then we sprinkle in just a little drop of the Cre recombinase. And so in some cells which get the Cre, the channel rhodopsin gets turned off, and the voltage indicator gets turned on. Whereas in the cells that don't get the Cre, they're only expressing the channel rhodopsin. So now we have mutually exclusive expression of either channel rhodopsin or the voltage indicator. So when we stimulate the cells with light, the ones with the channel rhodopsin will fire, but we can't see that. And we can only see postsynaptic responses in the cells that have the voltage indicator. Okay? And so this provides a way to quantify the synaptic connections in this culture. And this works. So here when we flash with blue light, we see, now these are EPSPs in this channel rhodopsin, excuse me, in this Archaerhodopsin expressing cell. And then if we add synaptic blockers, that response goes away. Okay. Now here we're stimulating the whole field of view. And so we don't have spatial information about which of these many channel rhodopsin expressing cells is the presynaptic cell and where these signals are going. So to address that, we've been developing systems for patterned optical stimulation. One of the nice things about using light to trigger cells to fire is that you can control when and where you deliver it. And so we've developed a system, many other people have done things like this too, where we use a modified video projector to stimulate the cells with different patterns of light in space and time. So we hacked a video projector just like the one that's displaying this image. Inside the projector, there's a chip with about 1 million individually addressable pixels in it. We use those pixels to modulate the blue laser beam on its way into the sample. And then we use flood illumination with red light to look at the response over the whole field of view. So each pixel, corresponding to each pixel here in this image, corresponds to one point in the sample. And it acts as an independently tunable stimulus node. So each one of these million pixels can turn the light on and off. And then the camera has about 1 million pixels in it. And so it has essentially 1 million independent recording nodes. Okay. And so just to demonstrate that this works, I took a video from YouTube and projected it onto a fluorescent film in the sample. So there's no cells here. This is just an exposure target. And so this is the view inside the microscope. And a single cell is about the size of the clown's head. So we can really stimulate in any pattern of space and time. Okay. So here's an application to neurons. Here are 2 neurons in culture. They're both expressing this optopatch construct, so they both have the actuator and the reporter. I've just artificially colored one green and one blue so we can tell them apart. Now we'll optically stimulate just the cell body of the one on the left, and I'll show you a movie at 1,000 frames a second where this neuron will start to fire, and there'll be an excitatory postsynaptic potential in this downstream cell. This movie is averaged over about 100 repetitions of the experiment in order to improve the signal-to-noise ratio. Okay. And it goes by quickly, so you have to pay attention. Okay. And so you can really see how the signal goes from the upstream cell to the downstream cell. Again, just to check that this is really synaptic transmission, we added synaptic blockers, and now you'll see the upstream cell will still fire, but if you look at the cell body of the downstream cell, you'll see no response. Okay. So this provides a comparatively higher throughput way to study synaptic transmission in defined cultures between defined pairs of cells. Now, we're interested not just in looking at pairs of cells, but also at large neural circuits. And this turns out to be challenging to look at a large field of view. The challenge is that we're running our camera at 1,000 frames a second. So this is a very short exposure time, which means we don't get that many photons in each frame of the camera. And so we need ultra-sensitive optics and very good laser systems in order to collect enough light so that we can get a signal in the short time. To do this over a large field of view is not something that can be done with commercial equipment. So through my company, we've developed a special microscope which can do this. And here I'll just show you an example of what we can see now. So here's the field of view. It's about 1 millimeter by 3 millimeters. So there's about 50 or so rat hippocampal neurons here. At a certain point in the movie, we'll turn on a blue light. Here we'll turn it on over the whole field of view. and we'll see the response to the cells. In this particular movie, we've added synaptic blockers, so we're looking at cell autonomous excitability here. There'll be a little icon which shows up when we turn on the light. This movie is at 500 frames a second, and it's slowed down tenfold so that we can see what's happening. So here's what we see. So now we can see every action potential in every cell in the culture as it's firing. It's a lot of fun to look at these movies. This turns out to create a big computational problem for us. The data comes off the camera at about 1 gigabyte per second, and so storing that and processing it is challenging. One of the big challenges we face is in image segmentation. So here, these neurons are all intertwined with each other and overlapping with each other, and morphologically, it's not so easy to segment them. And so we're working on developing activity-based segmentation algorithms. Some of them are borrowed from the calcium imaging world. It turns out they need to be adapted because of the different noise properties and different statistical properties of the signals in voltage imaging. Anyways, after doing that, we can extract these traces. So we can extract the firing patterns of the cells. Here's an example of a rheobase protocol where we stimulate the cells with pulses of blue light of 500 milliseconds long of successively greater intensity, and you can see how the firing patterns evolve under stronger and stronger stimulus. Something we're still trying to figure out how to do is how to interpret these patterns. We now have tens of thousands of these patterns, and to relate these to the underlying physiological state of the cells, the state of the ion channels and their metabolism, is something that we don't have any general tools for, and we're trying to figure out how to do in specific instances. Great. Now, one of the problems that we face is that these action potentials from the neurons are just so brief. They only last a millisecond. And even with the best cameras available, which can image at 1,000 frames a second, we can only detect these events. We can't really see the propagation through the cell. So for instance, in the movies I showed you of synaptic propagation, you couldn't really see the propagation. Basically, it looked like the whole neuron lit up at once. And so we wanted to find a way to map the motion of the wavefront through the cell. And one of my students came up with a very clever trick for how to do this. And I'll tell you how it works. We take a movie of a neuron firing. Here's an example at 1,000 frames a second. And if we look at a single pixel in this movie and plot the fluorescence intensity as a function of time as the neuron fires, we see the fluorescence go up and it comes back down. And we're sampling here at discrete intervals corresponding to the exposure times of the camera. But we have prior information about what this cell is doing. We know that it's producing an action potential, which is a smoothly varying function that goes up and comes back down. And so we can fit a simple spline interpolation to this discretely sampled data. And then we can set a threshold, and we can ask when does this interpolated function cross that threshold. And we can do that with a precision that's better than the exposure time of the camera. We do this fit independently at each pixel in the movie, and then we make a new movie where we subdivide each frame into many subframes, and we only show a flash at the time corresponding to the interpolated function crossing the threshold. Okay? So this is a model-based interpolation algorithm. A similar idea has been used in the single molecule fluorescence imaging field for many years. If you have a fluorescent molecule in a microscope and you look at it, the image of the molecule is blurred out by diffraction, and then it's sampled at discrete intervals by the pixels on the camera in the spatial domain. But people have known that you can fit the point spread function of the microscope to that blurred data, and you can localize the molecule in space with a precision much better than the diffraction limit of the microscope. This is the basis for many of the so-called super resolution imaging techniques, which are becoming very popular in cell biology. So we've essentially taken that idea from the spatial domain and rotated it into the temporal domain. Okay. And it actually works. Here's an example. Here's a neuron, a rat hippocampal neuron. We're optically stimulating just the cell body here, so there's no electrodes. First, I'll show you the movie at the native frame rate of the camera, so 1,000 frames a second. And this movie is averaged over a few hundred repetitions of the experiment, again, to improve the signal-to-noise ratio. Okay. So we stimulate the cell, and it lights up, and we see it fire. but the signal goes by too fast for us to see the propagation. The whole event only lasts 2 or 3 frames. Now we'll apply this interpolation algorithm to the data. And the thing to remember here is that we're processing each pixel independently. So we don't impose any wave-like motion on the data. Any waves we see are really there in the underlying structure of the data. So now I'll show you the wavefront propagation. This is interpolated to 50 microseconds a frame, so 20,000 frames a second. Okay? And here's what we see. Okay? So you can actually see how these signals propagate through the neuron. Here's another example. Here we're optically stimulating, again, just the soma. This movie was taken at 1 millisecond a frame, but it's interpolated to 10 microseconds a frame, so 100,000 frames a second. Okay.
Is that still trial averaged?
Yes. So these are all averaged over a few hundred trials. And all of the data is shot noise limited, and so we have to average in order to improve the signal to noise ratio. If we had a brighter reporter, we wouldn't have to average so much. Yeah.
Should I take it to mean that the action potential propagates faithfully to all the dendrites of this structure? Because that's not typically the case.
So these are
not rather like that.
yeah. So these are cells in culture, we have to remember, and so it's a relatively small field of view. So things that are very distal we can't see here. But indeed here the impulse is propagating everywhere we see. Now you could ask whether there are changes in the waveform as it goes, whether this is active conduction or passive conduction. And this kind of analysis doesn't tell you about that because we're just looking at the wavefront. If you actually look at the shape of the waveform in different parts, you do see some changes as it goes out.
So you said that in the super resolution microscopy, they actually have the point spread function of the microscope.
Yeah.
But
here we don't.
Hear you correctly, you're doing just a cubic spline, so you don't actually have a template. Does that affect things?
It's a good question. So we have a little bit less information here because we don't know the underlying function we're fitting to. It turns out in the super resolution business, you can actually use lots of different roughly symmetric functions and you'll get roughly the same answer. And the same is true here, but you have to pay attention to what it is you're fitting to. So we set a threshold as a certain percentage of the maximum deviation for each pixel. And so then what we're mapping is the piece of the wavefront at that threshold. Now, if you picked a different threshold, you might see something which looks a little bit different if the action potential is changing shape as it goes through the cell. And so you have to sort of pay attention to which piece of the waveform you're surfing along here. Yeah, so that's an important point. You have to remember that there's no free lunch. We're here looking at information at 100,000 frames a second while we're only acquiring at 1,000 frames a second. And so there's assumptions built in. And at the end, there's no more information in this analysis than there was in the raw data. This is a way of just highlighting very subtle differences in the timing between one pixel and another.
So you use a symmetric function to fit this time. So you need the pairs to know which side.
Here we're not using a symmetric function. Here we're using something asymmetric. And we fit not just the threshold, but also the sign of the derivative. So we look at the upstroke here. Yeah. Good. All right. Let me show you some more examples. So one of the neat things that you can do with this toy is you can stimulate different patches of dendrite, and then you can see how the signals go. So here's an example where we've picked just a little patch of dendrite up here. And you'll see something a little bit surprising when you look now at the action potential of propagation. So pay attention to where you see the signal start. We're stimulating up here. And it looks like the signal is starting somewhere down here. Okay. So what's going on? I'll show you another example. Here we're stimulating a patch of dendrites over here. And pay close attention to this little piece of spaghetti over here. So this is at 50,000 frames a second. So even though we're stimulating over here, it looks like the signal is starting over here. And so we hypothesized that we're seeing dendritic integration here, that the signals are coming from the dendrites, and that perhaps this is the axon. And we're seeing that at the axon initial segment, the threshold for action potential initiation is lower than elsewhere. And since we've set our threshold above the action potential initiation threshold, we're seeing that the above threshold part starts here and then is propagating down the axon and back into the soma and into the dendritic tree. To test this hypothesis, we took this cell and we immunostained it for anchorin G, which is a marker of the axon initial segment. So now I'll show you an overlay of the same cell where in red it's now the anchorin G immunostain, and then green is the rest of the cell. And so this is the biochemical data, and here's the anchorin G stain. And so indeed, we're seeing the signal start at the distal end of the axon initial segment. Okay? And so you can correlate this high time resolution functional data to the underlying biochemical state of the cell. And so that gives us some confidence that what we're seeing here is real and it's not just computational artifacts. All right. So now, what about in vivo? So far, I've showed you everything in cells in culture. As a warm-up to going in vivo in the brain, we started in the zebrafish heart. It's much easier. And so we made a transgenic zebrafish, which expresses this calcium and voltage indicator in its heart. So now we have caviar in our fish. We took the fish and we put it on the microscope. And here we're looking at the calcium signal in its heart. And as it beats, you can see the calcium waves travel over the heart. We developed a custom spinning disk confocal microscope for acquiring optical sections inside the heart of the fish. Fish have a two-chambered heart. Here's the atrium and here's the ventricle. This is now looking at the voltage signal. And so you can see the electrical impulse traveling over the heart of the fish. This is not single trial data. This is averaged again over a few repetitions. And then by taking many such images and computationally stitching them together in different z-planes, we made a three-dimensional reconstruction of the cardiac action potential. Okay, and so you can see the propagation. This is acquired inside the live animal. And then we developed a dual wavelength imaging system to look at the voltage and the calcium at the same time. So we're now working on translating this to mice. I should say we've tried to do this in zebrafish brain and so far have not succeeded there. But nonetheless, we decided to go ahead in mice. And we made a floxed mouse with this optopatch gene in it. And so by mating it with different Cre drivers, we can target expression to any cell type we want in the body. And so here's an example where we used a mouse which expresses Cre under control of the NAV 1.8 promoter, which is expressed in the dorsal root ganglia in sensory neurons. And so in the offspring, this optopatch construct is expressed in the sensory neurons. We then explanted those neurons and looked at them in the dish and optically stimulated them and recorded the fluorescence. And indeed, we can record optically from these cells. This is in collaboration with Humphrey Zhang at the Allen Institute. And here's what these cells look like. The nice thing about the transgenic is now every cell expresses, and the expression levels are much more homogeneous. So now, in principle, we can look anywhere in the animal and do optical electrophysiology. These are in cultured cells. Our local hero, Yoav Adam, who joined my lab about 1.5 years ago, has been working very hard and very productively to try to translate this to initially brain slice and then to in vivo. And he's had to deal with many technical challenges along the way, both on the optical front, because imaging in tissue, we have to deal with scattering and laser power issues. And also on the molecular front, it turns out that the trafficking of these proteins in vivo, for reasons we don't understand, is not as good as in the cultured neurons. So he's developed some new constructs which have much better trafficking to the plasma membrane. And just very recently, he got some expression in the hippocampus in mice, so using a CAMK2 Cre driver, and has shown that with optical stimulation, he can record optical action potentials. This is in an acute brain slice. And so now we're very excited to start zooming around the brain and doing optical electrophysiology in acute slice. Doing this in vivo is still a ways off. It doesn't work in two-photon imaging. Okay. And here, just to show that we can really look anywhere, we used a universal driver to express it throughout the whole animal. And so you can see expression in the cerebellum, and the dorsal root ganglia, and the heart, and the spinal cord, and in fact in all of the organs of the animal. One of the things that excites me about this is the possibility to do optical electrophysiology measurements, not in the brain, but in places where people just haven't really been able to do electrophysiology before, either because the cells are mechanically inaccessible or too small to patch, and so to look at unconventional electrophysiological effects in other parts of the organism. All right. The last thing I'll tell you about is an application of this technology toward disease modeling. So we'll go back in vitro now, but we'll apply it to human neurons. And so I've been working with my colleague Kevin Egan in the stem cells department at Harvard to apply this technique in human iPS-derived neurons. And so the idea is you can take a person, anybody, and take some skin from the person, and then reprogram these skin cells back through embryonic development into a stem cell-like state, and then take them forward down a different developmental pathway and grow them into neurons, which you can grow in a dish. And these neurons, they'll wire up and they'll start to fire, and they're genetically identical to the neurons in the head of the person who gave the skin. And then we deliver these optopatch genes to them, and we can characterize the firing patterns and the response to drugs, and then try to use that to learn something about what's going on in that particular person. And the motivation for this is there's a lot of interest in studying human neurons, particularly in disease models. And one of the bottlenecks in characterizing these cells has been the difficulty of functional characterization by manual patch clamp electrophysiology. These cultures are often very heterogeneous, and in order to overwhelm this variability, we need to look at hundreds or thousands of cells, and we'd like to look at lots of different conditions and lots of different people, and that's just not practical to do with conventional patch clamp. So as a warm-up exercise, we've been looking at ALS, so a neurodegenerative disease of motor neurons. And as with many diseases of the nervous system, just because we have a name for it, that doesn't actually make it a thing. The name just describes the symptoms. And ALS is very complex, like most diseases. There are many underlying genetic causes which can lead to similar symptoms. For about a quarter of the cases, the cause can be attributed to mutations in one of these genes. For about 3/4 of the cases, we don't know what causes it. And so this is part of the motivation, again, for using humans, is that in the mouse model, there's a particular mutation which is instantiated, but that might not be reflective of the diversity of underlying human causes. So we've been working on cells derived from a patient who had a mutation in this SOD1 gene. That's this slice of the pie. So it's a single point mutation in this gene which caused the disease from which the person died. So in Kevin's lab, they made stem cells from this person, and then they edited the genome to correct this single point mutation so that we have two populations of cells that are genetically identical in every way, except for the single point mutation which is causal for the disease. And then we can compare their electrophysiology in the dish. And so we deliver these genes to the cells, and then we optopatch them, we stimulate them with light, and we look at their firing patterns. Here's what the data looks like. This blue trace is an example recording from one cell. Here we're stimulating with 500 millisecond pulses of light of successively greater intensity as we go from left to right, so the cell fires more and more. We find every spike, and then here the data is arranged in a raster graph. So each row is one cell, and each spot is one spike. These columns correspond to the bursts of activity when we're stimulating the cells. And we've done this on hundreds of cells with a mutation and hundreds of genome-corrected controls. Then the question is there a difference between this pattern of dots and this pattern of dots? And of course, there's many sophisticated ways to look at this. We've started very simply just by averaging the rows together to look at the mean firing rate as a function of time during the stimulus. And after averaging thousands of cells, we found that indeed there is a difference. The mutant cells fire more in the absence of stimulus. So during these troughs where we're not stimulating them, you see the red line is above the black line. But under strong stimulus, the mutant cells fire less. You see here, the red line is below the black line. Red is mutant, black is control. Based on earlier experiments that Kevin had done with Clifford Wolf using manual patch clamp, they had inferred that this hyperexcitability in the unstimulated state was due to a deficit in a voltage-gated potassium channel. They attributed it to the Kv7 potassium channel. This hypoexcitability in the strongly stimulated state seems to be, is at least plausibly explained by a similar mechanism. If you look at the waveforms here, what we often see is that the cells saturate at a depolarized state. And so essentially due to a deficit in this potassium current, the cells are, we're getting sodium channel inactivation and the cells are depolarizing and getting latched at a depolarized state. That's our current understanding. Interestingly, there's a drug, retigabine, which can activate the Kv7 potassium channel. This drug is approved for epilepsy. And Kevin's lab has shown that adding retigabine to these cells restores the wild type firing pattern. We've seen that also in the optical measurements. And just a few weeks ago, GSK announced that they're doing a clinical trial of retigabine for ALS, and we'll see what happens. Okay. So before I finish, I'll just give one other comment. So we've been talking about the brain here, and of course that's the subject of the day. But one of the things that excites me about the possibility of optical voltage measurements is that we can look at electrophysiology throughout life. Every cell is encased in a lipid membrane, and in principle this membrane can support a voltage. And this voltage pulls on all of the proteins and all of the charged molecules associated with the membrane. And so we expect electrophysiology to be important in many situations besides in neurons. Here's a chart showing resting membrane voltages in a variety of cell types and membrane-bound structures. So here's the brain and the heart. Here are bacteria. Mitochondria, of course, have a large membrane voltage, which is used to power the ATP synthase. And for reasons that are not really well understood, some plants and fungi have gigantic membrane voltages, as large as -250 to -300 millivolts. And it's very hard to get electrodes into cells which have a cell wall, like plants or fungi, or into things which are really small, like mitochondria or bacteria. And so with these optical tools, we have a hope of starting to explore electrophysiology in these other situations. So I'll give you just one example of something surprising we saw when we looked at membrane voltage in E. coli. So people have known for a long time that many bacteria have voltage-gated ion channels, but nobody had ever measured the membrane voltage in any intact bacterium before, single bacterium, because they're too small to get an electrode into and they have a cell wall. So we looked at the membrane voltage in E. coli. We're not doing anything to them here. They're just sitting on the cover slip. This movie is sped up about 6-fold from real time. And here's what we saw. So we discovered that bacteria generate electrical spikes. And this was a big surprise. We didn't know what ions or ion channels are involved or what's the biology of this. Why are they doing this? And there are millions of species of bacteria in the world, and we don't know anything about the electrophysiology of any of them. So maybe someday we'll have a conference on bacterial days in Jerusalem. Okay, so I want to say something about these tools we're using. It's just incredible to me that we can take all of these genes, which have evolved under totally different circumstances, and staple them together to come up with new functions which have never been selected for. The Archaerhodopsin that we work with comes from right here in the Dead Sea. It was discovered 30 years ago. The Sha'ananim comes from this freshwater alga from the south of England. We fuse the Sha'ananim to a fluorescent protein derived from a coral, and we fuse the Archaerhodopsin to a different fluorescent protein derived from a jellyfish. And then we actually link these two proteins with a peptide derived from a pig virus. And it's just neat that you can staple these things together. And we can only do this because ecologists have been going around for decades classifying these seemingly useless organisms. All right. So here's the Dead Sea. I told you about these Archaerhodopsin-based voltage indicators. I told you about this calcium and voltage indicator, about this optopatch trick for optical stimulation and recording, about this algorithm for mapping action potential propagation with high time resolution. And now we're applying these tools in neurons and other cell types. And we're still working to get it to work in vivo. That's still a ways off. These are the people who did the work. I've had many great collaborators. Ed Boyden gave us the gene for Archaerhodopsin to get started. We did a lot of the protein engineering with Robert Campbell and Ed Boyden. The work in fish was with Florian Engert. In mice with Venki Murthy and Bernardo Sabatini. Making the transgenic was with Hongkui Zhang. And the work in stem cells was with Kevin Egan. This project was started by Joel Kroll, who was a postdoc in my lab. He really bet his career on this. He's now an assistant professor at University of Colorado Boulder. And then Daniel Hochbaum and Adam Douglas did a lot of the early work. And Yoav Adam has been carrying the torch and is trying to get this to work in tissue and in vivo. These people pay the bills. This is my lab, and I'm happy to take questions. Thank you.